So in the series of short videos, this one is on the structural components of DNA. This is the usual disclaimer about the video lecture that it is meant only for quick revision and educational purpose for the students who are affected by COVID-19. Okay, so as we know that DNA is the genetic material in all the organisms except some viruses, RNA viruses, which have RNA as genetic material. And it is a master molecule. It is considered as a master molecule because it not only stores, but it carries genetic information from one generation to the next generation. And structurally, it is a polymer of 4 types of deoxyribonucleotides, which are joined by phosphodiester bonds. So it's a polymer of 4 different types of deoxyribonucleotides. So structurally speaking, it is made up of 3 components. What are those 3 components? There is one sugar, a pentose sugar. Pentose means the one which is made up of 5 carbon. And that pentose sugar in DNA is deoxyribose. Deoxyribose. The second major component is phosphate groups. These two, pentose sugar and phosphate group, they serve the structural role. They provide the structural strength to the DNA molecule. Third component, that is nitrogen base. There are 2 groups of nitrogen bases which are present in DNA. One is called as pyrimidine, of which you have cytosine and thymine present in DNA. And the second group is called purine, in which adenine A and guanine G are present in DNA. And nitrogen bases, their primary responsibility is the storage of genetic information. Okay, so there is a good division of work among the components of the DNA. The sugar and phosphate group, they play the structural role, whereas nitrogen bases, they are involved in storage of genetic information. So pyrimidine structurally, they are made up of a 6-membered nucleus. 6-membered ring is there in the nucleus. So they have this structure in their nucleus, whereas purines, they have a rather big, rather bulkier heterocyclic 9-membered ring in the nucleus. So this is the nucleus, this is the ring which is present in the purines, that is adenine and guanine. Okay, so what is meant by nucleoside and nucleotide? So nucleoside means when you have base, when base combines with sugar, nitrogen base combines with sugar, the resulting compound is called as a nucleoside. Resulting compound is called as nucleoside. And when the nucleoside combines with phosphate group, the resulting compound is called as a nucleotide. So we must understand this. What is the difference between nucleoside and nucleotide? When base combines with sugar, it gives you nucleoside, any of the 4 bases. And when the nucleoside combines with phosphate, the resulting compound is called as nucleotide. Okay. So basically nucleotide means base, sugar and phosphate, all 3 together. So this is a nucleoside. For example, you have a base. This is a purine base because it is a purine ring. And this is a sugar. This is a ribose sugar, right? And the 2 are joined by a bond which is called as glycosidic bond. It is a beta glycosidic bond. This is a beta glycosidic bond. This bond is formed between N9 of the purine ring. This is N9 of the purine ring. And C1 of the sugar. This is carbon number 1 of the sugar. So that glycosidic bond, which is a beta, which is in beta configuration, it is N-glycosidic bond, which is formed between N9 of the base of the purine base, purine ring, and C1 of the ribose sugar. So these 2, nitrogen base and sugar, these 2 combined, you get a nucleoside. Take another example. This is sugar. Okay, but this is deoxyribose because at carbon number 2 you don't have a oxygen atom here. You have an oxygen over here, but you don't have over here. So this is deoxyribose. This is ribose. This is deoxyribose. And this is a pyrimidine nucleus, pyrimidine ring, right? And again, the bond is formed between N1 of the pyrimidine ring and C1 of the sugar. The bond is same, beta glycosidic bond, okay, but it is formed between N1 of the pyrimidine ring and C1 of the sugar molecule. And what are nucleotides? Nucleotides are esters of phosphate esters of nucleosides. Nucleotides are phosphate esters of nucleosides. And most commonly occurring nucleotides, the common site of esterification of phosphate group is carbon number 5, C5. C5 of the sugar. This is C5 of the sugar. Hydroxyl group of carbon number 5 of the sugar is the most common site of esterification of the phosphate group. So phosphate group comes and binds here. Okay, a bond is formed between the phosphate group and hydroxyl group of C5 of ribose. And so resulting compound, this one is called as a nucleotide. So you have a sugar, which is a deoxyribose sugar. Okay, because there is no oxygen over here. You have a nitrogen base. Here it is a purine ring. And you have a phosphate. So these three together, when they are together, the resulting compound is called as a nucleotide. Okay, so when the sugar is ribose, then when it combines with the nitrogen base, the resulting compound is called as a nucleoside. And when nucleoside combines with phosphate, the resulting compound is called as nucleotide. But when the sugar is deoxyribose, then the resulting compound, when it combines with base, the resulting compound is called as deoxynucleoside. And when deoxynucleoside combines with phosphate, the resulting compound is called as deoxynucleotide. So in DNA, we have deoxynucleotides, whereas in RNA, we have nucleotides. Okay. For example, for DNA, so let's say nitrogen base is adenine. So when it combines with the sugar, that is deoxyribose, so resulting nucleoside is called as deoxyadenosine. And when it combines with phosphate, the resulting nucleotide is called as deoxyadenylate, or you can call it as deoxyadenosine monophosphate if there is only one phosphate group, deoxyadenosine diphosphate if there are two phosphate groups, or deoxyadenosine triphosphate, that is DATP if there are three phosphate groups attached. So when the nitrogen base is guanine, when it combines with sugar, deoxyribose, the nucleoside you get is deoxyguanosine. And when it combines with phosphate, the resulting nucleotide is called as deoxyguanylate, or you can call it as deoxyguanosine monophosphate, or deoxyguanosine diphosphate, or deoxyguanosine triphosphate. Similarly, for cytosine, when it combines with sugar deoxyribose, the nucleoside is deoxycytidine. And when it combines with phosphate, what you get is deoxycytidylate, or deoxycytidine monophosphate. Similarly, for thymine, when it combines with sugar deoxyribose, the nucleoside you get is deoxythymidine. And when it combines with phosphate, the resulting compound is called as deoxythymidylate, which is a nucleotide, deoxynucleotide. Or you can also call it as deoxythymidine monophosphate, diphosphate, or triphosphate, depending upon the number of phosphate groups attached. Thank you.